autism.
Yeah.
But the autism is so extreme that he's extremely, and I was extremely,
Yeah.
like he was raised Christian, so he's
Kind of
like,
sad.
he was like extremely homophobic. A lot of the trolls like got into pretty awful stuff.
Kind of sad.
It's a mix of like a lot of factors. I don't recommend looking it up here.
Yeah.
It's a mix of a lot of factors. He's not a good person. The trolls aren't good. Well, okay, not all the trolls are bad. Some of them are pretty good.
Sorry, should have done that before I started it. That was in my head.
It's fine.
Okay.
Some of them are pretty funny and didn't entirely like hurt him. There's one that was like as a joke, basically saying, "I'm the real Christian."
Yeah.
This one's a fake. And somehow Christian actually thought that this person was impersonating him, even though it looked like nothing like Christian. Obviously, it was a joke. But he took it so seriously that to prove that he was a real Christian, he literally doxed himself.
Oh my gosh, that's sad.
Like the other person that was pretending to be Christian as a joke wasn't even trying to do that. He was just being funny. He was not trying to get Chris to dox himself.
Yeah.
On his phone.
Yeah.
Because he's that stupid.
I don't know. I just think it's sad.
It's like sad and funny.
Yeah, I mean, I guess. I don't know.
Some factors are funny, some factors are sad. Some factors are just really gross and messed up. It's a lot.
Oh, all right. Hey. Ooh. Thanks. Full screen. Oh, okay. We're into the theme. Whatever. I think it's just as good as it gets. How are we feeling, guys?
Good.
Yeah. Oh, whose notes did I find sitting out? Somebody left their notes out. I don't know. Maybe
Not me.
maybe they're not here. It was these ones.
Do we really need to know all that for like? That's cute.
No. Somebody who's not here.
Do we really have to know all that just for the lab exam?
What do you mean?
Like, do we really need to know all that?
Grace. So if you were to ask literally any professor that question, you need to know the entire textbook and everything we ever talked about in the lab. That's what every professor would tell you. So y'all have to know those study sheets. You do need your lab coats today. You will get points off on your exams if you do not have your lab coats. Yes. If you forgot it and it's like actually far away, I have, the extra ones and I don't mind, but if we have a lot of people that didn't have, it's going to be first come, first serve.
Like I know most stuff in the Kahoot. Like I would able to answer most of the Kahoots to myself. I think I'll do fine.
You should.
It's just like I look through like the worm names and stuff. And I'm like, do we for real need to know all that?
Yeah, I'm going to ask you about it on the test.
Uh-oh.
So I'm just saying, there's a reason I tell you to know it, right?
Ask,
I am going to ask you about the worm name.
Do the dippy thing.
I'm going to ask you one name, not all of them, right?
Oh, just one.
One of them, but not like, is this a worm? It's going to say, well, how does it do something? I'm not going to tell it now because it's on the test. You guys are literally about to take it, but yeah. Yeah, if you don't mind, I'm sure. Do what you need to. Look through the study sheet. That's the thing that's going to help you the most with it. I double checked every single question I had on the exam. I wrote it like the other day, and then when I printed it out, a couple of the questions weren't on that study sheet, so I actually changed them to questions that were on the study sheet. So I've double checked it.
Study sheet is thing with the PCR directions and stuff?
All the pictures that I posted for everybody about the lab study sheet. Yeah.
Maybe I might be stupid, but yeah, I'm stupid. I'm gonna start asking. all
Yes. Who's asking me? Okay. Okay. Yes.
When you're counting,
Mhm.
it doubles. So you've
Yeah.
gone from 1, 2, 4, 8, 16, or 2, 4, 8, 16, 32.
Well, they'll go to 32. Yeah, it'll go one on the
Okay,
first
start with
cycle.
1.
You go from 1 to 2. On the
Okay.
second cycle, you go from 2 to 4. That's what you get at the end of that cycle. Does that make sense?
Yeah.
Yeah, I figured that's what you're asking, like the sequence of.
Yeah, I just get confused. Do I start with 1 or do I start with 2?
Yeah, you'll start with 1 and the one cycle. If I were to ask how many do you get from one cycle, you would say you have 2 at the end of one cycle.
18. Yeah, 1, 2.
Anybody have any other questions? Logic? Yes. Huh? Yeah, the plasmid. What about it? So that was, I don't know if you were there that day, because you missed a day, didn't you? You didn't? That was the one, so that was the day we had the little Tupperwares with the plasmids in them. Okay, so those little circles were plasmids. That was P-glow. Does that help you? Okay. Okay, anybody else have any questions?
No, every single time there's like an exam, I'm still literally just going through the class. And do we really need to know
All of this.
Do we really need to know
Everybody, every semester, at least one person asks me that.
Really?
Yeah. I would not be wasting my breath if I didn't think you guys needed to know it, believe me. Are you guys feeling like no, absolutely not. But if it helps you even just a little bit, that's the goal. Yeah. I just met, if you guys know Rhiannon, she's the nursing advisor. She came to my office to basically figure out what, she doesn't talk to us very often, right? Even though we are the prereqs, all the science people anyways. And people find they have a more difficult time with the science classes than with anything else. And so what she hears on that side is, people complaining about each class. And, we'll talk about physiology and my class in particular being like just really hard and, nobody can do this and whatever. And, but I think people in the class kind of would think about, yeah, it's hard, duh. But people who are complaining about to her are complaining about the people teaching the class. And so would you like somebody else teaching this class? So that's what I'm saying is like, I don't want to say, I don't know how to put it into words, because I didn't hear the complaints, but that's what was implied in the meeting. So she was pretty sure that they were full of crap. So she was going to see each of the professors and see like, okay, so what are you doing, in your class for your students? Because they're making it sound like it's just hard and they have to like figure it out on their own. And I'm like, oh yeah, let me tell you what I do for my students. Yeah. Let me show you what I do. So I am not saying, and right. There's always, and I'm not, you might be the person listening to me talking right now. You might be that person that will just insist that this is my fault, that this is a hard class for you, or that you end up failing or only getting a C or something because I didn't teach the class or whatever, and might write that on somebody did that on whatever that, yeah, right. Somebody who claims to have gotten a B made a similar comments to that on Rate Your Professor or something. I guess they said that I didn't teach anything at all. They had to teach themselves entirely on their own, and they got a B. So they said I was appallingly unprofessional. And I might be like colloquial, right? I might be chill, I might be casual, and some words might slip out of my mouth. But last time I checked, that's everybody. Period. Yeah. And I might not be as professional as some of these stuck up other professors that other people have. But I feel like, wouldn't you rather I talk to you like equals?
Yeah.
So I'm not saying, for all I know, this person was just mad and they're just BSing and whatever. That is what it is, right? But so we always have at least a couple people every semester, but I would hope that you guys would appreciate, I'd try to do a lot for you. I'm not bragging or anything. I'm happy to do it for you. That's my job. But I hope I give you enough resources that if you guys are studying, you're like, oh my gosh, this is a lot more resources. And that's your problem. like to picking which resource to use, as opposed to like, how am I going to do this on my own? You know, and that's the goal for me, giving you as much as I can so everybody has a choice of what works for them. And then at that point, you know, it just ends up being how much work you decide to put into it and then what grade that you get. I would hope at the end, whatever grade you get, that you feel like you earned it for whatever reason you earned it, right? So if it's not good, then at least you would look at it and say, I didn't work hard enough. And I would hope that, you know, that I gave basically because I hope that I'm giving you everything you need. That's what I'm saying. Yeah, that 4.4 used to be 5.0.
I took this course during the 2023 fall semester. I'm unsure what was different about this course than others, but the unprofessional behavior was appalling. Questions are not encouraged during lectures. I had to teach myself that. Really?
Questions are not encouraged in lectures. You see what I mean?
We'll get in there.
I was about to say, yeah.
You guys put what you think about me and that's fine. But when I saw that, because I check on it, I very rarely check on it, like maybe once a year. But when I checked on that and I saw that and I was like, bullshit all day long. Like, no.
I wish you could boo them.
Yeah, me too. I wish I could boo them also.
You should have a
Dr. Nephew were my favorite
Right.
professors.
Okay.
And like, because you understand me really well, despite us only knowing each other for like 8 semesters. I was like,
Well, that's my job too. I'm supposed to like accept and work with everybody. If people don't ask for help, it's hard for me to do that. But that's part of it too. And what I talked about with Rhiannon was that as well, that if you're not asking for help, I can't help you.
if being unprofessional is like what you want in OSU OKC, I don't know what to tell you. It's literally
Like, yeah.
like no one is going to be professional.
What are you expecting exactly? So anyways, I actually kind of pride myself on being on the level as opposed to being snobby. So whatever. Anyways, so let's start with this. This is not as long as you can see. Your slides are like probably 61 slides or something. Mine's longer because I have my notes in between. I'm not going to go through every step of the notes this time, but I'll go through each section and then show you my final work. Okay. But yeah, so we'll go through this. Antimicrobial drugs. Antimicrobial means any drug that could be affecting any microbe, not just bacteria, not just anything else, right? It's going to be a blanket term when we say antimicrobials. If you're trying to design one or pick one, you'd rather it be microbicidal. You want it to kill instead of, you know, inhibiting. We prefer that. And we've already seen that antibiotics, some of them are microbistatic instead of, right? We've seen that in our own things. So it can vary from organism to organism. We want it soluble in body fluids because we're putting it in a body, obviously. And that it's going to hang around long enough to do its job and all this stuff. So the top one, the reason I kind of skipped that one, toxic to the microbe, non-toxic to the host. So that's actually that concept. It's obvious, right? We want to kill the bad guy and not hurt the host. That's going to be a term. There's a term for that we're going to get to here in a second. So I just want you to be aware of that concept first. All right. What else do we want our drugs to do? They want to help what your body already should be doing so that your body can still like make an immunity or something like that. And it's not doing all of the work for it. We want to, yeah, we already said remain active in the tissues for a long time so it can work. Be delivered to the site of infection. Some drugs, so if you're trying to treat an infection that is like on the skin by taking an oral antibiotic, you need to be sure that antibiotic is actually going to travel to the skin. If you are a diabetic and you have poor circulation in your limbs and you stub your toe, and this happens all of the time, and you get a wound, and now we need to treat it with an antibiotic, but that antibiotic can't get to that area because there's not enough blood flow to it, so we can't get enough of it there. That's why people lose limbs. And if you guys don't believe me, this is a common thing. Go talk to Becky about her dad. He just lost all of his leg gradually.
Who's Becky?
Becky Dye. She's the girl with the, I don't know. She has the dark short hair. Yeah. She's petite. Yeah. Yeah. Becky with the good hair. Yeah. She's in my Tuesday morning class.
It's very passionate.
Yes. She's a hard worker. She really is. Yeah. Just to be clear, she's the one that I drug a Tulsa for. I told you guys about that, right? Yeah. Yeah. She's got a family problem going on out there. Anyways, that's a lot of personal probably info that be supportive. So yeah, so we want to make sure that we're choosing the drug that's going to do the job that we need it to do without harming the person that's taking it. So some terminology to be aware of: prophylactic. These are things to prevent. That's easy enough. I don't know. You want to prevent a disease. You might start taking antibiotics before you go into a surgery, for example. So you already have it built up in your system, especially if you're getting like a hip replacement or something. That'll help prevent biofilms from forming. Antimicrobial chemotherapy is just a fancy word for drugs that kill germs. That's all it is. Antimicrobial is germs. Chemotherapy, anytime you hear that word, doesn't always have to do with cancer. It's anytime you're putting a drug in somebody, that's chemotherapy. It's a chemical therapy. That's all it is. All right, antimicrobials, like we said, it's all inclusive. It's a blanket term. And antibiotics. So if you see antibiotic and we think biotic, okay, that's, all the living things that we're trying to kill, and that's not correct. If you see antibiotic, look at the B for bacteria. Antibiotics treat only bacteria. Antibiotics do not treat fungal. Antibiotics do not treat protozoa or helminths or virus. They treat only bacteria. All right, so where do we get our antimicrobials from? So some of these are natural in nature. Some organisms just make them as part of like their defense. We know that that's true because we know the story about penicillin. It was mold, and then they realized that mold was preventing the growth of bacteria, and then we turned it into medicine that you could use. So semi-synthetic is whenever we are altering those drugs that already existed in nature and putting like extra groups on them or something to help them work better or differently. Because if you're resistant to penicillin, then maybe you won't be resistant to amoxicillin that we altered. Okay. So synthetic are the ones that we just make from scratch. They might be based on like the theory behind those drugs, but they are something that will be synthesized from scratch. The narrow spectrum, these are ones that are going to only target like a specific group. Let's say endospore forming bacteria, for example, or mycobacteria, even that specific, whereas broad, obviously, that's going to target a lot more. Could be all bacteria. Could be gram-negative bacteria, whatever. Okay, so like I already said that part, that it's made by other organisms. We know it's fungi can do it, but bacteria can also make antibiotics against other bacteria. So it's a useful defense mechanism. They want to protect their stadium seat in the world, if you will. So they make those to keep the other guys away so that they can have their spot and be left alone. It's usually to help them maintain their spot. So like we already kind of went over this. There was another slide. I already mentioned these, but semi-synthetic, changing it to make it more effective. Synthetic is designer drugs, designing it specifically for a purpose, but they usually are going to be unsuccessful at permeating the membrane just because it's a lot to design and have that be able to cross a membrane. So that can be a problem. So let's say you are ready to start your treatment. These are the three things you need to know: the identity of the microbe, the degree of its susceptibility or its sensitivity to various drugs, and then the overall medical condition of that patient. So yeah, I am giving you a little bit of a review for some of the stuff we're going to have on the exam. So first, basically summarizing this: what is it? What can we use to treat it? And can the patient handle it? All right, so the first step of that was, what is it? So we need to figure out what it is before we start treating the person, unless they're in a life-threatening situation. Then you might just throw a shotgun at them, several drugs all at once. If somebody comes in with meningitis and you suspect it's meningitis, you're not going to wait for any results. You're going to just shotgun antibiotics at them. And then when they get the results back, you'll just pull out all the ones you don't need because you don't want them to die in that time. And we kind of talked about that a little bit. You can use whatever samples. We'll talk about diagnostics in chapter 18, so we don't talk about it too much here. One of the things we can do is test for the susceptibility in the lab. And we learned about this. This was the Kirby-Bauer test. So you can create your lawn, stamp your antibiotic discs on there, and then let it grow. Your zone of inhibition is the area where it didn't grow. So growth is inhibited there. If I show you a plate that has a zone of inhibition on it, we can't say whether it killed or it just was static, right? We can't tell the difference up front, but we do know that it did inhibit growth somehow. So we can say that at least. So remember that maybe, I'm just saying. Antibiogram is just determining which of these are going to be effective and like how much you would need for them to be effective. So of course, there's your Kirby-Bauer test. The larger the ZOI, the more susceptible we are. And we see this guy out to the side, kind of out here. That guy is, and this guy down here too. Vancomycin. This thing is completely resistant to vancomycin, which is kind of concerning, but and whatever that other top one is. But anyways, it gives you that idea, and we'll measure our zone of inhibition in millimeters, right? And then use that information to determine that. This is a similar thing. This is an E-test, and it is essentially the discs. But we've put it in a strip where at the top of the strip is our highest concentration of that antibiotic, and down at the lower part of the strip is less and less and less and less. So you can see, like on this image here, where this is the growth here, and down here at the bottom here is the concentration that is the minimum amount needed to inhibit. So that just tells you just right there, you don't have to do any math or anything. So that is what, there's a term for that, the minimum inhibitory concentration, the MIC. So that will just tell you that without having to calculate it. We don't need to calculate it, that's fine, but it exists. We already said this. Minimum inhibitory concentration is just the minimum amount of that drug that you need in order to inhibit the growth. It says it in the name, it just sounds like it's a lot. It's not actually. So we can use that information. The smallest amount that you need to prevent that microbe from succeeding in life, and then using that to treat somebody. If we do a test like that and the treatment still fails, that would be for 3 reasons. The inability of the drug to diffuse into that area. So that's number 1 down there, our summary little, it can't get there, just like we were saying with our diabetics. Number 2, resistant microbes in the infection area. That just means like when you did your sample and you grew the things on there, we didn't pick up the resistant microbes, or it became resistant as it was growing on the patient and we just didn't happen to test it. If you're just taking a swab, there might be other parts of that wound that have resistant microbes on it already, and we just didn't get them. Same thing with that bottom one. It could be more than one microbe. And the one you were testing was just an area where there was just the one, or the one is the only one that grew in your sample for whatever reason. So those are all things that could be the reason you would have to go back and try to figure out which one is going on. If it's not working and your patient has diabetes, it's probably the number 1. Easy. But how do you figure out the 2 and the 3? You're just going to have to do more samples, probably do more cultures. Our therapeutic index. So this is another term to be aware of for sure. It's a very important one for understanding how to pick our antibiotics for treating our patients. This is essentially the ratio. It says the ratio of the dose of the drug that is toxic to humans to its effective dose. Basically, the toxic dose versus the effective dose. So the dose where you take it, where it starts harming you, and then compare that to the dose where you take it that it inhibits the microbe. If those are very close together, you need 0.5 to kill this guy, but at 0.5, it also causes toxic effects. That's dangerous, right? You might want to pick a microbe where those are farther apart, right? So yeah, that's all this is talking about, is that it is just a lot of words to explain that, but yeah. You want to prefer drugs that are less likely to be toxic, of course, but it also matters about resistance and things like that. We have to consider all of this. So we also have to consider these things. Any pre-existing medical conditions that can influence how the patient responds, either to the toxicity of it or even to how effective that antibiotic could be, just depending on blood flow and things like that. History of allergies to certain drugs, penicillins and sulfas are going to be the more likely ones that you'll be allergic to. Underlying liver or kidney disease. And we'll think about for a minute why those specifically. We're not asking about underlying heart disease, are we? Interesting. We're asking about literally liver or kidney. And then we also have to look out if somebody is an infant, an elderly person, or pregnant, because they've got a whole lot of other things going on with their bodies that we have to consider. If they're still growing, some of these drugs can affect that. Some of these drugs are going to inhibit nucleic acid synthesis directly. So we don't want to give that to an infant who's growing, right? Drugs that you have taken, in fact, probably do. You just didn't know how they worked. But anyway, so the liver or kidney, the liver is the most likely one that's going to be like processing the drug itself. You take a drug orally, and usually in the liver, that's where we're going to be like breaking it down into a metabolically active version of it, or just processing it in general, removing it from the other things that are in a pill. Because I'm sure you guys know this. When you take a pill with an antibiotic, it's not just only antibiotic in that pill. It's like things to stabilize it, and maybe even like so that it doesn't just oxidize in the air and all sorts of stuff like that. And package it in a way that it's going to help it get to whatever part of your body it is. And the liver often will help deal with that. It processes a lot of drugs. It's like its most important role that it has is processing like toxins and things like that. Next, we have kidneys. Kidneys are going to filter for us. So if we get that drug into our body and through everything, if it comes to the kidneys to get filtered out, and we're making urine and everything, though that's pretty sensitive tissue, and it can cause damage in that tissue. And then if it can't filter out toxins from your blood, you build up toxins in your blood. And that eventually is going to lead to kidney failure when that happens. It's like a cascade effect. So it can be pretty dangerous. So that's why we worry about those specifically. Yeah, there's some other things we could think about. Synergistic and antagonistic effects of drugs. Drugs can interact. That's all this is saying. Some drugs can help. Some drugs might inhibit. And then, of course, we need to think about our biome. Our biome is there for a reason. We need it taking up the stadium seats in our body so that the bad guys can't sit down. That was that term that we learned back a while ago was microbial antagonism, right? So that's where we have our good guys taking up the spots so the bad guys can't sit down. They can also like use up the nutrients or even produce toxins to get rid of them. So we don't want to get rid of those because we need it. So how are we going to choose a drug that's going to not affect any of that? And unfortunately, there's not a perfect drug out there. It's just not. So let's think about this. Selective toxicity is that term for what we were talking about earlier. We want to kill the microbes and not harm the host as much as possible. We want to keep it in that mindset, right? We want to target things in the microbes that we do not have. For example, penicillins target peptidoglycan cell walls. So easy target, man. We don't have that in our cells, do we? We might have it in our biome, so there might be some risk there. But as far as for our actual tissues, hey, we're good with that, right? So that's one really great target to think about. How we're going to, so here we come back to this, y'all, because we saw this in our last post with our control agents. These are our antibiotics, though. So we have the same four: the cell wall, the membrane, the protein, and the nucleic acids. And then we add on one more: folic acid synthesis. So it's the same four plus folic acid. This is a little chart that you guys can come back to reference. Don't worry about anything on here that isn't on your study sheet. So you just, if you come back to this, just go ahead and cross out ahead of time the ones you don't need to know. I could have done this, but like y'all, I literally put this together at like 11 o'clock at night. So, so bear with me here. So yeah, so I don't know what else to say. We do, one of the things we do need to know about our drugs, we do need to know what part of the cell they target. So those 5 possible things they can target, that's one of the main questions I'll be asking you for the antibiotics. Which of the following choices affect the cell wall? Which of the following choices affect the membrane? Or what part of the cell does penicillin affect? And I'll give you all those choices. So that's a very common one for me to ask on this, because the exact mechanisms by which they work, I don't care about, and neither do you. And so that's why they're not on your study sheet, okay? But know the basics. All right, effects on humans. These are talking about side effects. Like we said, if it's going to be affecting protein synthesis or nucleic acid synthesis, we do that in our cells. So we have to be concerned, and we also have membranes, right? So we have to be concerned about the effects that that could have on our cells. So they could cause damage, they could cause development of allergies, or they could completely ruin our biome. We want to avoid that. The drugs can serve as antigens. So remember, antigens are anything that the immune system can react to. And then when you see it as an antigen, you develop antibodies against it. And then every time that you see it, antibodies are going to come out and attack that drug. And that can lead to anaphylactic shock if you're having that response. We'll talk about that. That's a chapter 17 thing, y'all, about how that works, but it's coming. But yes, so it can be either from the drug itself or from, they say, metabolic alterations. So as the body's processing it, some intermediate in there, you can be allergic just to that. So if we could maybe change how that is processed by changing the chemical itself, we could avoid that in people. Obviously, I told you guys, penicillins and sulfas are going to be the ones that you're more likely to be allergic to. That's an obvious given. All right, superinfection is a term that we need to know as well. And we already know how this works, actually. This is when I give you antibiotics, and then it clears out your stadium seats, and then yeast can overgrow, or C. diff that has endospores can overgrow. And that's superinfection. So stuff that's already in your body that we clear out a whole population, and then the other thing what's left can grow, and it can cause problems, disease, because it shouldn't be in that amount. I went to go visit my mom while I was in Tulsa, and my dad was out of town, which is fine because she needs to be like her, but by herself sometimes, and talk to people that are not my dad. And it's sad to think that that was an exciting visit or whatever, but like she's one of those people that's like, she's kind of agoraphobic. And I don't mean kind of, I mean, she's very much so agoraphobic. She has like safe places that she'll go, like this particular grocery store or this particular gas station that are safe places. And she doesn't even like to have to go to those. So she is at home a lot. Anyways, I went to go visit her, and she's been having GI issues, and she's been diagnosed with diverticulitis. And that's where you get these little pockets in your intestine, and they like trap fecal matter and the bacteria in them, and they'll kind of grow unhealthy and cause inflammation in those pockets. And she's been having big issues with it, even though she's been following like their rules. She's not eating the things she's not supposed to and everything. And so they started her on 2 different antibiotics. I don't remember what, yes, I do. I do remember the other one. It's 2 that we're going to talk about today, actually. So they started her on 2 different antibiotics. And I'll tell you about why and what they are in a little bit, because it actually applies to, I feel like, a lot more people than you would think. So, but yes, so I feel like that was kind of pertinent, and we'll get into that story in a little bit. She's doing all right. She's just could be better. They like remodeled their home. So they had a tornado hit their house, and it only like knocked off the roof. It was a 2-story house. So it's like everything on the top floor had to be completely redone. And they're like, why don't we just redo everything? So like replaced their whole kitchen and their bathroom. And so it's the first time I'd seen it since they did a lot of the new big renovations. But anyways, it's just kind of cool to get to see. She chose like this green paint for her cabinets. And when she asked me about it, I was like, like, I don't like this. And that's saying a lot, because my house, if you guys saw it, I have black walls, and I have like dark purple, and I have like dark green, and like dark ceilings. And like, I'm that person. Okay. Like that. What do they call it? Like goblin core is what they call it. It's like the witchy plus cottage core feel. And I have skulls and all of that. That's me. Okay. So that's saying a lot when I think that a color is too much, but I went, and it was actually it was good. She did a good job. So yeah. Anyway, so we want to kill the bacteria, but we don't want to be toxic to the host. All right. So remember, antibiotics only treat bacteria. Selective toxicity. This is my notes for what we just talked about, by the way, if you can't figure that out. Selective toxicity, kill the microbe without hurting the patient. Therapeutic index, toxic dose versus effective dose. Super infection, kill off the good guys. The bad guys will overrun. And then the Kirby Bauer, I don't think I need to go over that with you. I hope I don't need to go over that with you. So this is a chart showing the spectrum of activity for a large number of antibiotics, not all of them. These are groups of antibiotics, some of them are. And then what like organisms that they would treat. But like, look at tetracyclines down here. Like this is not a very good color. Tetracyclines down here. That is pretty impressive. They also, so you'd be like, why not just treat everything with tetracyclines? Well, it's pretty toxic. That's why. Okay, so let's get started. Now we're doing the thing. Okay, we're going to go through the drugs like we did with the methods. And I'm going to break them down into the wall and the membrane, all of that stuff. Okay. Starting with the wall, we have 3 antibiotic groups that we want to be aware of. The first one is the beta lactams, AKA the penicillins. So most of these are going to be penicillins that fall into this category. Those are like our big starting points. Anything psyllin. That is pretty self-explanatory. They have a beta lactam ring, which I'm not going to have you draw or identify or anything, but I would be aware that that's associated with affecting the cell wall. It's in your study sheet too, but like, look, this square, that's beta lactam rings. Okay, cool. So what are these going to do? Oh, it doesn't even talk about it. Whatever. Okay. We're going to inhibit the cell wall. That's what we're going to do. Next ones, cephalosporins and carbapenem. So again, cephalosporins, that's a group, and it works pretty similar to the penicillins. It does have a beta lactam ring. It's just not the true original beta lactam psyllin. It's like a derivative, and it is has been altered to make it work better. And then we have the carbapenems. These are our last line drugs. These are only used if you are going to be resistant to things that you think are pretty strong acting things, like vancomycin. So we will use carbapenem as a last line of defense. So if something is resistant to carbapenem, they're probably resistant to all of the other drugs before it, and then that's a problem. So yeah, there are other ones that are not on your list. These also target the cell wall: bacitracin, isoniazid, and vancomycin. Bacitracin is one of the 3 antibiotics that are in your Neosporin, the triple antibiotic cream. So bacitracin, neomycin, and I can never remember what the third one is. Always forget it. It says, I'm not going to test you on it. And then isoniazid treats tuberculosis. Oh, it already says it down here. I should have just looked at that. Whatever. So here's what we have so far. That's all you really needed to get. Cell wall, we have the beta lactams, and all these can kind of fall under this in a way, but they're all going to affect the cell wall. Okay. I'm not going to ask you if carbapenems are also beta lactams, but I just be aware that beta lactams affect the cell wall. And here are some more specific groups that also affect the cell wall. Okay. The psyllins, cephalosporins, and carbapenems. All right, next we have the protein synthesis affecting pathways. We're already there. All right, this goes a lot faster than the other one did. We have aminoglycosides. This is a group. Okay, so this is not an antibiotic. We have antibiotics that fall into that category. Okay, these are pretty broad spectrum, and they come from those organisms that I'm not going to test you on. These guys are aminoglycosides, and you're looking at this and you're like, okay, there's tiny words on here, and you're correct. But one that you do need to know that is on your list here, and that is affected by stopping the ribosome. It's down here, and I'm going to circle it. It's with another guy that's related to it, and that's tetracycline. So you do need to know that one. So it affects the ribosomes and makes it so that I'm not need you to know the details of how it affects the ribosomes, but it does affect the ribosomes, and therefore, if it affects ribosomes, it affects protein production, right? That's translation. So that's how that's going to work. So that is tetracycline and glycylcycline, y'all. So yeah. All right, tetracyclines, they are going to be derived from streptomycin and other things that might be related to it. It's derived from that. So we added other groups onto it to make it more effective. What do I say about it? Blocks ribosomes. That means it blocks that. The macrolides. I don't care about the structure of these. I know it has all this BS about the structure. Look at it, and we do this and that with it, and whatever. What you need to know about the macrolides, they affect protein synthesis. Okay, I'll even write this on here so we can just all see it. Affect protein. That's what this whole category is about, right? Synthesis. And then also about these guys, not every mycin fits into this category, but a lot of them do. Like we already saw vancomycin affects the cell wall. That's a different thing. But most of the other ones will fall into this category. So this is going to be most of the mycins. And I'm not testing you about vancomycin, so you don't even have to worry about it. So good with that, right? So just think the mycins belong in this category, the macrolides. That affects protein synthesis. We're already on to the folic acid. Folic acid is something that you need in your body to create nucleic acid nucleotides. That's what I'm trying to say. The nucleotides that build up your nucleic acids, that's their primary function. And they're also involved in amino acid synthesis. It's a really crucial role in the ability of the cell to survive. We, as human beings, do not synthesize folic acid. We get it from our diet. So it's another one of those things like our vitamins that we need provided to us that makes it a growth factor, right? So those would be growth factors for us, but for bacteria, they don't need that. They're just going to make their own. So that makes it a good target. We can target that because we don't have that pathway in our own bodies. We already talked about cell wall, obviously, good target. This also, none of our cells are going to have this target, right? I'm sure you have noticed, though, what drugs fall into these categories. Our best bets, the ones that work for us and are less likely to make us toxic, the penicillins you can be allergic to, and what was the other category? The sulfas. Isn't that lovely? The ones we would want to use most. So these are our sulfas or sulfonamides. Sulfasoxazole, I'm not going to be good with these words either. You guys who are presenting on this chapter, I'm sorry. But yes, I'm not going to ask you what they do, but here they are. Okay. Sulfasoxazole, that one, this one here. Sulfur, I can do other words. I don't know why it's the S's with this one. Silver sulfadiazine, and then we had trimethoprim and sulfamethoxazole, which we talked about in lab. That was our SXT. So these are all going to fall into, remember, trimethoprim is also going to fall into folic acid area. They do the same thing. They just work together in different ways. These usually are going to be either competitive or non-competitive inhibitors. And some of them might work together, like one's competitive and one's non-competitive. And we're just going to double dose it and make it work stronger. So these are pretty effective because of that. That was it, man. All right, the things that target DNA and RNA. So this is usually synthesis of these, okay? We'll start off with the quinolones. That's a group, but more importantly, the group name that you need to know is fluoroquinolones. These are just a group of antibiotics that affect the synthesis of DNA or RNA. They're very, very potent, very broad spectrum, and also very toxic, obviously, because we need DNA and RNA also. So these guys can cause seizures and other brain disturbances because the cells in your brain are a little bit more sensitive to that than other parts of your body. And it doesn't have the ability to heal. So that can be a problem too. This usually how they work, and you don't need to know this, is by preventing the untwist, like your DNA is twisted. Yeah, it's a helix. So helicase is the enzyme that untwists you, and it prevents that from working. So if you can't untwist it, you can't open it up and you can't do the rest of the stuff. That's how it works. You don't really need to know it, but I'm going to give you some examples that you do maybe need to know. I don't need to know them directly by name. The more famous one on here is, of course, going to be ciprofloxacin. This is Cipro. So yeah, when you guys are sitting there saying, oh yeah, I took Cipro for blah, 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 or that person took Cipro, or levofloxacin, that's another one that's commonly used. These guys inhibit your DNA synthesis as well as the bacteria that you're treating. So now you know. But those are the ones I would know. These, the biggie, just for remembering the fluoroquinolones, are the floxacins. And hey, they both have FL. Fluoro, even though it has a U in it, you could just say it's like FLO. I'm going to make you spell it. Okay, next thing is the membranes. There's only one I need you to know on this one. And this is the one that I couldn't remember on the triple antibiotic, right? Yes, it was, because I already said neomycin. It's a mycin, so it falls in the protein. So this attacks the membranes. So when you get a triple antibiotic, you get bacitracin for the wall, you get neomycin for the protein, and you get polymyxins for the cell membranes all at once. So that's why it's so good at helping you heal. But anyways, polymyxins, they're pretty toxic to the kidney because we also have cell membranes. So we don't want to take these for long term. Again, they're also pretty narrow spectrum. So you want to be sure that you're, picking the right thing. That's the only one you need to know for cell membranes. You might want to treat biofilms, and this isn't on your study sheet, but biofilms, you have to be aware of like its levels, right? There's like thickness of different kinds of bacteria involved. The best bet is just to prevent them from happening if you can, but if you need to treat them, you might need more than one antibiotic, but daptomycin is one that we often use to treat biofilm infections if they do develop, and it affects the membranes, but yeah, I don't know what else. You don't need to know it. So there you go. Prevents quorum sensing, which is them talking to one another. So here's where we are right now. That's all of, that's everything. That's the drugs. Anyways, because we're going to talk about after that, other things. Oh, the other ones that aren't antibiotics. That's right. There's other things out there, guys, other than bacteria that we have to treat. Darn. So these are the bacteria treatments, okay? The floxacins, ciprofloxacin, that affects the DNA and RNA synthesis. Yeah, we already know Cipro, right? That's one of the antibiotics that my mom was prescribed. So we're going to come to the other one in a moment. And I said one of the other antibiotics she was prescribed, and I said that was all the antibiotics. And now we're going into not antibiotics technically. One of these can double. So that's why I'm saying that. And it's kind of important to know it, but we're not to it just yet. So fungal infections, if you have a fungal infection, this is a problem now. We're getting into treating eukaryotic organisms, and you are eukaryotic. So it's harder to avoid toxicity with these guys. But so we have to find some aspect of these guys to attack. And I don't need you to know what the aspects are, but I do need you to know your options are going to be amphotericin B. Yes, it does happen to be a macrolide, but I don't need you to know it. And then the azoles. And tell me you guys don't know the azoles. Ketoconazole, fluconazole, miconazole. I don't know that bottom one. Clotrimazole. But the top three I do. And fluconazole is very commonly prescribed for like yeast infections and things like that. Very commonly prescribed. And if you go to the doctor, not the doctor, if you go to CVS and you have a yeast infection, you want to use like Monostat or something like that, a lot of times that's based on miconazole. Thank you. If you have other kinds of fungal infections, they'll usually reach for ketoconazole. I had to take that when I had that sinus infection, then I had the fungal one, and then that didn't work, so I had to go to amphotericin B. They don't usually give that, like, it doesn't get to the sinus cavity very well. It doesn't deliver there, so I had to have a special apothecary had to mix it up for me, and I had to go spray this weird mixture that I kept in the fridge up my nose. It was the worst. I ended up having to have surgery anyways. It was a waste of my time. So next we have the so all you need to know for the fungals, azole at the end, except there will be one that is not a fungal one that ends in azole that I'm about to tell you about. But and what was the other? Amphotericin B. It's just on its own. Now we're on to the antiprotozoal and the antihelminthic drugs. We're going to start with the antimalarials. This is the history of where antiprotozoals came from, and this was quinine. Now, quinine it's available at very, very dilute quantities in tonic water. Back in the day, it was in much higher quantities because people were trying to not get malaria. And this kind of came out from the, British people trying to migrate into other people's countries and take over, like India. And so they had to figure out ways to prevent everybody getting malaria. And so they have quinine, and it was a very bitter tasting drug. And they had their little tonic water with their quinine, but it was terrible. So they came up with this cheap thing that they could mix in with it that was made of juniper berries. It's an alcohol made of juniper berries, made specifically to mix with quinine so people would drink it. You guys know what that alcohol is? It's gin. So that's what gin was originally made for, to get people to take their quinine. It's made from juniper berries, if you didn't know. Originally. But anyways, cool story. Now a lot of the stuff that came after that was based off of quinine. And then we have this other drug at the bottom. You don't need to know that is cool. That's ACT, whatever. So you don't need to know it. You need to know quinine and its derivatives. And they end in quin, Quine. So quinine and its derivatives. Next, we have the other protozoal drugs and the antihelminth drugs, okay? Right, so this is it, right? I told you it was coming, the azole one. So this is used for protozoal infections, metronidazole. That is not a fungus. So this is the one azole that got away from us. I don't know if this helps at all, but I feel like it says Metro on it. So to me, it like went to the big city. It went to the bigger, badder eukaryotes. And that's why it is in this category. But it's also it's just an amoebicide. It's used for a lot of other protozoal infections though. And then it's also good for anaerobic bacteria. It doesn't say that on here. I think it's on a later slide, but still, we use it for anaerobic bacteria as well. Let me write it on here because I otherwise won't remember to mention it later. Anyway, so my mom was put on ciprofloxacin, DNA inhibitor or whatever, and metronidazole. So that was her antibiotic. So they're trying to treat probably an anaerobic bacteria. And you guys know what they probably think it is? Clostridium difficile. That's like the #1 anaerobic bacteria that would be causing problems. And it's hard to get rid of because it's endospore forming. So that's why we have 2 antibiotics on board to try to help with it. So they think she has C. diff, I guess. Anyways, so that's cool. Then we have the anti-helminthic drugs. There's 4 of them here, but the 2 that I know best and have heard of the most are praziquantel and ivermectin, just because we see these in like veterinary situations a lot. So especially like it's commonly used, ivermectin to treat parasitic worms in general, but especially like heartworms. And if you have a dog, or maybe, I don't know if you guys treat your cats for heartworms, but if you treat your dogs for heartworms, you might have like those chews that they have every month, and that has ivermectin in it. And so they just keeps ivermectin in their system so that if they encounter the worms, and they do, they can't grow. And I say they do because this is Spaghetti and Meatball. You guys haven't got to meet him yet. My night class got to meet Meatball. Yeah. Yeah, the difference, some people are like, how do you tell the difference? Well, Meatball on the right, he has a little tuxedo on. He has the little white on his front and his little white paws. But anyways, and you can see little Spaghetti's face at the side of that picture. They can't be separated. They'll cry for each other if they are. But when we adopted them, we knew, this was back in like February, we knew they had heartworms. So we were told up front. And it takes dedication. So we had them for a month. They said, just have them for a month. They have heartworms, and let them get comfortable with the home and you and everything like that. Because then you lock them in a cage, and they get high doses of ivermectin. And then they have to get locked in a cage and not allowed to run around or anything. They only got to go potty on a leash like once in a while, and they weren't allowed to run around or even walk around or anything. Because if they got too much of their blood pumping too hard, the little pieces of the worms, the dead worms from the ivermectin, will get caught up in and cause basically embolisms and cause them to die. So we had to be really careful. And so we did it though. We made it the whole way through, I think it was 6 weeks that we had to do it. And we made it through, and now they're running around like banshees or whatever. So they're good. We had them double checked. Like we had them checked, of course, right after the treatment, and then another 6 months later, just to be sure they were clear, and they were clear. So we're really happy that they're still doing good. They're cuties. All right, so antivirals, these guys will either keep the virus from getting in, block penetration, usually how the spike protein attaches, or we will block replication, transcription, or translation. And all the stuff that we've been talking about with the cells, but remember, viruses are not cells. So if we're going to block their replication, transcription, and translation, you need to be targeting maybe something that the virus makes that helps it do those things, because you don't really want to target that machinery that we have in our own cells, because you'll be hurting our own cells. So it gets really, really tricky with antivirals. When we talk about something like HIV, and we've already mentioned that HIV has all of like these specific little things that it can do. Those are great targets. But unfortunately, once you get the HIV into the DNA, nothing is going to help with that. So it's just, you can keep the virus at bay, but it's still going to be embedded in the DNA. Anyway, so a lot of these will be based on that virus. And we can also stop the maturation of the viral particles. It's usually referring to like stopping them from budding. So as they're trying to bud out and wrap themselves in a membrane, they have to pinch off somehow. We can prevent that from happening. All right, we'll start with the ones that prevent getting in. The only ones that you need to know for that one is Tamiflu and Relenza. Tamiflu and Relenza block influenza's neuraminidase action. So we know flu has H whatever one, N whatever one, right? So we know what H and N. The H is hemagglutinin, N is neuraminidase. So we did talk a little bit about hemagglutinin, but now we're on the neuraminidase side. This guy helps the virus get in, and it also helps with the budding out. So, or helps, it stops it. That's what I'm trying to say, right? Prevents those things. So the drug, I'm gonna like, now the drug prevents it, but now, yes, neuraminidase helps it get in. We're gonna block it with Tamiflu and Relenza. That's what I'm saying. Jeez. Cool. The ones that affect nucleic acid synthesis, we have, I don't know why it's not on the slide. I had a slide that had acyclovir on it, but I don't have it anymore, but it is on your list. And I think it's a pretty important antiviral to be aware of what it is. So I don't need you to know any more details other than it affects the DNA side, the nucleic acid side of things. And acyclovir, if you haven't, maybe people know it better as valcyclovir or Valtrex. So this is treatment for herpes treatment, and it can be used for other viruses as well. It's a pretty effective one. This is what you get if you guys get the Abreva or whatever on cold sores. It has Valtrex in it, basically, valcyclovir. So it works pretty well. Cool. So here's what we have so far with our fungi, our protozoa, and our antivirals. How about drug resistance? This could be due to genetic versatility and adaptability of these guys. They multiply a lot more than we do, right? I mean, there will be like hours and you have billions from just one. It's ridiculous. So there's a lot of opportunity there for mistakes to be made in the DNA. Sometimes those mistakes can be beneficial. That is literally how we've been taught evolution works, right? Natural selection, survival of the fittest. It's not that the organism is deciding to have these helpful things. It's that the ones that have the genetic mutations that help them survive, just randomly, they survive and pass on the gene. So it's not like there's them choosing or something like that. It's been, it just works that way. It's the way DNA works. Whether you think it was designed that way or not. However you think it happened, that's how DNA works. So drug resistance, it's going to be either intrinsic or acquired. Acquired makes sense. You're going to acquire drug resistance. You don't have drug resistance, and then the cell next to you gives it to you through your little sex pili, or you pick it up from outside of you, or you get it from a virus. Cool. We get it. Intrinsic, though, that means you already have it. This is not meant to be complicated. Intrinsic drug resistance means that if you make... Hey.
Oh, hello, everyone. So sorry.
Yeah.
I apologize.
You're okay. Intrinsic, yeah, like I know we're not over time at all, so I just want to be sure that I wasn't doing something wrong. I sometimes move into this room with my other classes because I can't get the computer over there to like, I hate that room. 100 is my least favorite room. Yeah, it is. Yeah, the internet is really bad, so sometimes I just get sick of it and we just come back in here. So sometimes I don't know if I need to be in here because I think I might be taking up the room when somebody else is supposed to be in here, but all right. So anyways, intrinsic, that just means if you make penicillin, you have to be resistant to it. That's all it means. So if you're going to be making it to protect yourself, then you must be. And a lot of this stuff came from bacteria. So antibiotics. Spontaneous mutation is what I was just talking about this. I do have a video on Canvas. I was going to play it, but I can later if you guys want me to, but it's pretty cool. It's just demonstrating the fact that like bacteria just divide so much that they're at some point, because they divide so much, statistically speaking, they will make the helpful mutation just because they have so many cells. So at some point they will make that helpful mutation and be able to have resistance, and then they can spread it to other bacteria. From there, this can be just because of normal mistakes that you make. in your cells, even our own cells will occasionally make mistakes when they're replicating. But especially so in bacteria and viruses, they do it kind of loosey-goosey because they like occasionally getting those, mutations. It works to their advantage. Not really as much for us when you're multicellular. You need them all to work together. But so you could have that happening just because, or also because of mutagens causing mutations. So that's anything that would force you to have genetic changes, like a lot of carcinogens would do. UV, for example, right? Or the other one, the ionizing, scary one. All right, so we had the spontaneous, or you can have horizontal gene transfer. Where are we getting that from? That's going to be conjugation and transformation and transduction again. That should be familiar to us by this point. But transformation, picking up the outside DNA. It's the whole experiment that we learned about that we're going to have tests over here in lab in a minute. Transduction is when bacteriophages, they do their thing by inserting their genes into the bacteria. Remember, that was like lysogenic conversion. And then we have conjugation, which is the sex pili that are going to transfer the DNA between cells. And we also have something new on here, which is transposons. This is when we have genes that are able to essentially jump from a plasmid into another plasmid or from a plasmid into a chromosome. We only really see this in bacteria, but that's transposons. Isn't that something? We see gene transfers are actually extremely frequent in nature, and even our own biome can have a part in developing the drug resistance. So how would this work? How do you become drug resistant? Well, I don't need you to know all of the details, but these are the possibilities, okay? You could get genes that make it so that you have enzymes that break down or inhibit the drug from working. You could stop the drug from getting in, so it's permeability or uptake, so make it so that it's not as easy for that drug to get taken in or stop the cell from taking it in. And then we have eliminating the drug entirely if it does get in. So they'll like shuttle it back out, which is nuts, but it does do that. Then we have just like, instead of preventing the access, literally just reducing the points that it can access. So less likely for that drug to get in and cause toxic effects to that bacteria. And then changing metabolic pathways. So that folic acid pathway that they need to go through to make folic acid, it's got several steps in it. They can just find ways to go around it. Like it shows down in the bottom. Oh, that drug acts at the B to C point. We'll just work around it. It's fine. So that's how they become resistant. The mutations let that happen. And then all you need is like one bacteria, just one. In this case, it has like maybe two just for visual effect, I guess. Let's say you have one bacteria that gets a helpful gene. The person takes the antibiotic, but that one survives. Now all these seats are open and it can just take up those seats with resistant guys in the environment. And then it can also sometimes shuttle its new genes resistance to other bacteria. It's a big problem, right? Video that I was going to show you, if I have time, I'll show it to you, but it's literally like, I think I've told you about it. It's a whole like table auger plate. And we start with E. coli and we just grow it for like 48 hours or something. And at each point after that, we have increasing concentrations of antibiotic, and it's just normal E. coli with no resistance at all. And within 48 hours, it's growing already in that middle, because at each stage, it just gets a new mutation, because you just need to grow enough to get that mutation to survive. And that's all you need. And that's terrifying. That should be. All right, some other things we got problems with: our role, human role, and how we developed this. Getting prescriptions for antibiotics, even if you have a viral infection. I think we mentioned this before. You have a sniffy nose, a little runny nose, and maybe a scratchy throat, and you're feeling a little bit down. They're like, okay, here's your antibiotic, and just send you on your way. I'm like, you got a cold, here's your antibiotic. And then so many people will come to me and be like, yeah, but my doctor prescribed me antibiotics for my cold. And I was like, oh, then he's the problem though. So just saying. So the people can get over colds just fine on their own. Just let them have a chance. If they start having actual inability to get over a cold, then give them treatment with something else, like an antiviral. But you don't need to ever be giving somebody an antibiotic if they do not have a bacterial infection. This is laziness, is what that is. Because now you're exposing all the bacteria that are in their whole biome to that, and any of them can become resistant and then spread it on to pathogens. So that's a problem. Shotgun, that's just giving a whole bunch or high doses of antibiotic when it's not needed. And then similar kind of thing, bacteria can just adapt to that. And then other countries that we are giving our antibiotics to, selling them to or whatever, they are misusing it and not being aware of that. That makes sense. All that stuff does, right? All right, next we have the hospital factor. We have a lot of people who are already there that are weak, they're sick, everything like that. So they're more likely to get infected. And they're also probably going to be on antibiotics already too. So now we have all these bacteria that can get exposed to antibiotics because it's all there, and then they can become resistant. Sometimes we have poor hygiene or PPE practice, so universal precautions, people not washing their hands or changing their gloves. It just happens sometimes. It's not always somebody doing bad on purpose, but it just happens. And all you need is one transfer. And now, because of stuff like this, we have essentially 100% of all staff ever seen in the world at this point now are resistant to some kind of antibiotic. That's not natural. They don't come that way. So thanks, world, I guess. It's terrifying. We already know about MRSA. MRSA is not the worst of it, not by far. We also have in animals, 80% of all antibiotics in the US are given to livestock. Read that again. 80% of all antibiotics in the US are given to livestock. Why? They're not sick. Yep, it's for meat. They say that it helps them grow bigger and stronger and be better products or whatever, whatever. But it's the same antibiotics, or they might work the same. And then their bacteria get the resistance and it can pass on to humans. And it happens all the time. Ridiculous. Okay, this is the problem. We're entering probably, maybe not in our direct lifetime, but we're getting very close to it, a post-antibiotic era, meaning that we cannot treat things with antibiotics anymore because things are just going to get resistant to the point that it's not good. So these are the biggest threats, the worst threats. You already see on here, I've already told you that if you are carbapenem resistant, we've got a big problem. So here we have carbapenem resistant Acinetobacter. It causes a lot of a variety of disease, GI kind of stuff usually, but there's that. It's just some random thing. It's completely resistant now, cause random stuff. Candida auris. This is not Candida albicans, which is like normal in your body. You can have auris around too, but this isn't like we usually get with yeast infections typically. If you have a lowered immune system, you might have Candida auris overgrow. And then when you do, and you try to treat it, but you have a lowered immune system, you could potentially become resistant. And it can, and then we can't treat it. So that's a fungus though. C. diff now is becoming very, very resistant. It was already pretty resistant because it's just an end spore. Carbapenem resistant Enterobacterales. Those guys are like gut bacteria, like E. coli and stuff like that. And then drug resistant Neisseria gonorrhoeae. When I say drug resistant, I mean we have strains of gonorrhea that aren't rare now that are completely resistant to every antibiotic that exists. That's what I mean by post, antibiotic era. We can't treat it now. And people ask me all the time when I say that to them, they're like, well, then what do you do? Like if somebody comes to you and they have gonorrhea and it's the resistant one, I mean, what do you do for them? Send them home and I tell them to use protection. What can you do? There's nothing you can do. Unless you want to put them down or something like that. No, I don't mean that seriously. But yeah, no, there's nothing you can do once it gets to that point. So anyways, there's other ones on here. This is horrible. These ones are bad. We've got MRSA on this list. Cool. And other ones, if you want to look over this, if you're interested, these are a little less scary ones, but we need to find a new way of treating is basically what we're getting down to. Yeah. We need to use some way of treating infections that the bacteria potentially, hopefully cannot become resistant to. That's what we need to be looking for. Something reliable like that. So we could keep on going how we're doing and trying to find new drugs and then redesign so that they're not resistant, but they're just going to become resistant to those. So the better bet is to approach different things like using nanomaterials. You can build what you need out of these nanoparticles to do what you need them to do. And the likelihood of something being resistant to that is like 0, but it's a matter of making it work. Antisense RNAs, finding a section of the DNA of the bacteria and matching it up. And now its DNA can't work the way that it's supposed to with opening and closing and everything like that. Blocks it, can't reproduce. CRISPR technology, cutting out genes and stuff like that. bacteriophages, using them to insert crap into the genomes. You would have to target specific bacteria, unfortunately, but yeah. Things that attack specific structures on them. Other things that are good though, ways you can help your body out and prevent infection from happening. Probiotics are great. They are good. I highly recommend do them. Okay. Yes. Help your good guys out by having more good guys support them. If you have issues with your biome, these guys can support that. You can't replace your biome with these unless there's a whole lot of a procedure involved in doing something like that. But they can help supplement if you keep up with them pretty regularly. And it makes a huge difference if you're regular with it. So you can replace some of your lost microbes, but not all of them with these, but they can help with that. So we'll supplement our biota. They do work. Prebiotics are things that will, it says encourage the growth. It's oftentimes like food that these guys like, encourage those guys. So do it. Fecal transplants, sounds the worst. If you have C. diff, they might treat you with even stronger antibiotics than what my mom is taking and want to clear out your entire intestinal biome to blank. And then they will take poop from somebody that you're close to, usually spouse or something like that, and mix it up in a little liquid. And then via colonoscopy, while you are completely sedated, put it back into your colon and let that populate. So you don't, you don't have to see it or smell it, but yes, you would know what had happened. But that's the best way to get a new biome.
On Netflix, they did a series called Gut Health,
Mhm.
and they're like, don't do this unless you talk to a doctor. Basically, it was like talking about that, how your body's getting rid of the bad bacteria that it really needs to
Mhm.
survive. And it was talked like one lady was almost at death's doors, and she started doing her brother's
Poop.
poop, putting it in pill forms, like blending.
Yeah, you can do that. You can take it that way. Yeah.
Breathing it, and she started being able to eat again and all kinds of stuff, but she started getting his acne.
Oh yeah.
And then so she did her fiance, which then she got his depression. Like
That's crazy. Yeah.
she went back to her brothers. But I mean, between life, like to me, that's like fascinating.
So before anybody thinks that, okay, yeah, sure, depression from the bacteria. Yes, absolutely. Yes. That's a real thing. So a lot of mental illness now they're tying to your gut not working properly. It's not breaking down your foods and nutrients the way that you need it. And so it's actually creating, or at least playing into the development of mental illness. This is true. They have actual medical studies on this.
When was this found out?
I don't know, not that long. I mean, it's not that long ago, but not that far. Like,
Because I've never heard of that. That's crazy. I
like
thought it
within the
was-
last 10 years, I'm pretty sure.
I always thought most mental illnesses were just like
I didn't watch it.
an imbalance of neurons. And stuff.
Yeah, but I'm telling you that like your gut health can cause that. Yeah. Just based on like what nutrients that you get from it. So
Antibiotics when you don't need it. So for a while, the gynecologist and I both thought that I might have had like a UTI or vaginosis. So when the test came back negative, she's like, well, you probably don't have it, but just in case, I can still offer you antibiotics.
yeah. Yeah. Not until it's like really an issue, I would say. But anyways, this was my resistance thing, and I had just made random notes, like probably like a lot of you guys do. So random notes here, but then when I went to reorganize everything, I literally like essentially copied and pasted the areas that I wanted and put them into this is my note sheet for this one. So this is not as fancy as the other one. And like I was saying to you guys, it doesn't have to be fancy. It's whatever works for you guys. So yeah, I posted the other one online, by the way. The chapter 11 one is on there. So it's better than this one, but it's on there. Anybody have any questions about this or any last minute questions before the lab exam?
Through my notes one more time.
I just don't want to imagine her doing one. Yes. Yes. It's called
This is worse than number 2 pencil. Actually,
It probably does. Most of them do.
why